So the topic is herbarium. Let's share some PPT. First of all, let me give the small introduction about the herbarium. Herbarium, a plural herbaria, it is a collection of preserved dried plant specimen. The specimen housed at the University of Melbourne herbarium are predominantly dried and pressed. So the collected plant material dried and pressed and preserved. That specimen called as herbarium. And not only dried plant, and sometimes these herbaria also the stored in wet plant in the form of specimen. It is preserved in 70% ethanol. So this is the herbarium. So maybe herbarium mostly actually the collected in the stored in the form of specimen, or in the stored in the form of dried plant. So herbarium specimens are an important record what plant grew wherever time. And also it is useful for identification of new plants also. They may have been produced as a voucher for an environment survey or systematic research. This research act parameter recorded allowing anyone to go back, check identification, resample, repeat the research. So already you collected the plant that is stored in the form of specimen or in the form of herbarium. But whenever you necessary, you can check the same similarities. So there are four main aspects of making good herbarium specimen. First step before the steps of the preparation of herbarium, let's give just brief introduction about the kinds of herbarium. You know already general herbaria. The general herbaria are the different scale, international and national local. Already given the name itself, international herbarium. These are very large herbarium offer with more than 4 million specimens and have global representation of taxa. That means the specimens collected from different countries. Most of these herbaria were founded early in the history of formal taxonomy, the 1550 to 1800, and have grown to their present size over the years. So already name itself the worldwide. That means the almost on the global level, all plants are reported or stored in the form of herbaria. The largest herbaria found in Royal Botanical Garden Herbarium Kew. A national herbaria, these herbaria consist mainly the plants of country, only for the restricted to only country. For example, in India, only these are reported in Indian region only. In England, so only England restricted area. As far as possible, the representative of all taxa, different family genera of the country in these different type of taxa stored in worldwide, that is international. national level, in country level, that is national. And in our India, the Central National Herbarium, Howrah, that is largest one. Local herbarium, that is the separated to the restricted area inside the country, such as state, maybe district, even small area like nature reserve. For example, we can already we collected some herbarium from Dr. B.R. Ambedkar Open University. That is one local herbaria. And Usmania University also have the herbaria. Telangana have herbaria. They are the local. The local herbaria are useful production of local flora, district flora. They contribute to national flora and much help in catering the needs of the local educational and research institutions. Example, Southern Circle Herbarium of BSI, Coimbatore, only for Coimbatore. BSI Botanical Survey in Uppal, some India, Hyderabad also one of the herbaria present. Now, the collection types. There are the four main aspects to make good herbaria specimen. First of all, collect the plant material from the out natural area. After collecting, press and preserving, mounting to the paper and label it. So these are the main steps. First, let's see the collecting. What to collect? When collected plants for herbarium or water specimen, there are two basic points to remember. The two basic points are, first of all, whether the plant are in vegetative stage or in reproductive stage. First, you take part, include all available parts of the plant. That is the reproductive structure such as fruit, flower, buds, as well bark, leaves, juvenile copies, foliage, etc. Anything you can collect. That is one. So mostly we can collect the with the flower, the reproductive part mainly with the flower, twig, flower, branches. Second, include detailed notes about the plant and its surroundings. Not only collecting and also the note down the points, what you collected, where it collected, and time of collection. These are all. Date and time. When in the field, it is good idea to collect two samples of the plant. One for dissection and identification, and other for herbarium specimen. Herbarium specimen. The while in the field, always record details in the plant in our work file field notebook. Do not rely in our memory. This information will later be included in the specimen label. You already collected and label it, write the name and label it. This is useful for further. Uh, in general, aim to collect first of all at least the terminal parts of the aerial shoot, including leaves, reproductive parts, that is first collected. A representative sample do not simply choose to biggest or pretest only the simple portion, only simple small twig you will have to collect. More than one specimen from single plant if plant variable. Actually, what we have done so far for for preparation your herbarium, you just pick up one twig. But if there number of different characters are there, you can uh, pluck with one only vegetative part, only flower parts, and vegetative with flower parts. You can collect in different modes also. So this is more than one specimen from single plant. If it is variable, that means any the copies leaves, the base, adult leaves, higher rep. This will be given the same collecting number. Each herbarium specimen consists a sample taken from single plant. And specific collecting recommendation to different plants. For example, herbaceous angiosperms, grasses, herbs, separation different. Woody plant separation different. Ferns different. Algae lichen different. So I will explain later how the herbaceous are stored, woody plant stored, ferns stored, algae like and mosses are stored. And second one, field notes. In field notes, without detailed, accurate information, herbarium specimen is almost useless. Already you collected the plant, but you did not record. You did not record the information. Without information, how can you recognize in future purpose? So you should write the information of the plant. Uh, Next, what you write in field book, use waterproof pen or pencil so your notes are not lost the rain and mist. That is number one. Write your contact details in the front cover so the book can be returned to you if lost. So that is also, of course, it is a simple thing, but you should follow. Stick a copy of herbarium label in the back cover to remind you what information is required. Use one page per specimen so you have to plenty of space if you need to information later. That means you ensure the empty place so that you can write the information on the paper. So this is the field notes and numbering and tagging. Record the number are used to identify different herbarium. For example, you collected the 101 plants, you numbering it 101 plants and their information. The information means the plant scientific name and plant family, plant characters. So the best numbering system is the simplest. Start from one and continue the rest of your collecting career. First start with one, second, third, fourth, fifth, like that. Use your initial in front of the number to different your collection from someone else. Already, for example, the hibiscus plant collected you and also your friend, and some other also collected hibiscus. But hibiscus plant, when you collected, you should write your within in your initial. My initial Y Kumar. Y initial. Now the name indicate with Y dot hibiscus or hibiscus also you can write in short manner. Next one. After after collecting and labeling it, the specimen preserved in the field. So here given the mesophytic plant and those delicate petals built several one picked. This result to poor quality herbarium. So when it is tender, it is not useful, not optimal to preparation of herbarium. The best, but not necessarily most practical way to produce good quality herbarium specimen of these types plant is take your plant press into field. Deliberately press the specimen as you collected them. As this is not always practical or possible. Labeling specimen with jeweler's tags. Then storing them in sealed plastic bags out of the sun is okay on cool days. Maintain the specimen quality especially. The keep inside the plastic bag humidity by spraying the small amount of the inside the bag. Dump in some folded hand towel and place this bag in this plant. Put the specimen filled plastic bag or cooler like this. After press this like charts. Special collecting requirements. Already told you in beginning the tender herbaceous plants, woody plants, and ferns, grasses. When collecting fern, it is essential correct fertile fronts, fertile, with the spores on the under surface. That is important. If you collect only vegetative part, it is not useful. When not only leaves, but sporophyll leaves you should collect. That is the fertile cones with along with the cones. And small plants such as grasses, herbs collect entire plant. As it a minute small structure, you can collect the entire parts rather than twig. You can collect entire parts. Root, rhizome, tubers, inflorescence, all fertile. The entire plant. If the plant is small and it is good idea to collect multiple plant. This can be given the same collecting number, put on the same sheet herbarium card. If you collect the more than one plant, but you did not, it doesn't mean paste on different papers, different herbarium sheets. Even you collected the more in number, the paste on the same paper with same number. Number also same, one number. The collecting shelves and trees, in addition, general point, collecting it also good idea to collect bark either from trunk or ground, but make sure the note where it came from and how far up tree. This is what trees only, the biggest is eucalyptus like that. If the plant shows different leaf morphology due to things like juvenile and adult maturation, coping due to fire, drought, collect different types of leaves and notes, where these leaves came from, why you think they are present. The simple. For example, if you take the neem tree in the first stage when it is seedling stage and different, and after mature, that means after 10 years also it is different. When after flowering, after fruit formation also different. That means you can collect without, then in juvenile stage you can collect, and vegetative stage only collect. After a mature stage also you can collect in different stages and you collect at the step trees. And collection of the bryophytes and lichens. Collect entire plant. When the plant is very minute, you should collect the entire portion. If they grow closely together, include algae, old frost, and bryophyte sporophyte. These type plants are dried in paper bags, envelopes, not plant press, not a plant press, not plant press. So can be placed in bags and enveloped straight away when collected. Collecting fungi. Fungi are very fragile, so be careful when collecting, transporting, storing them. If the fungus is parasitic, remember to also collect some host specimen. Without host specimen, the fungi could not survive. When collecting a macrofungi, an entire specimen, including the stripe and root-like mycelium is required, such as agaricus. If there is more than one specimen growing in that area, collect two or three, showing different stages of development, right? Fungi deteriorate markedly upon dying. Accurately recorded field notes are essential. Photo taken prior to collecting can also invaluable of plant identification. For more information, fungi, see the fungi. No need, no need. Loss, no need. Ethics and environment, no need. Healthy and safety. Preserving and pressing. Preserving plants. Dry them quickly. Attend to specimen as they dry. Plant press like this. Plant press come various forms, but usually consist of two wooden boards like this. Here you can see in diagram. Two wooden boards. The wooden boards with 30 to 45 centimeters. Cardboard and newspaper arranged like sandpaper. Wooden board, cardboard and newspaper arranged like a sandwich. The straps are belt are wrapped around the press. Let it be in several days. Careful drying. No need. Specimen preservation requirement. Harvest and deliver specimen with care to prevent them from breaking part of the falling off. The following above the should quality specimen. One is succulent or fleshy plants or fruits, aquatic plant, mosses, liverworts, and fungi. So these are the special preservation. Preservating for succulent. The succulent fleshy plant, it is easily infected with fungal damage and require additional care. To prevent mold and hide drying, change the paper more regularly to use more absorb the paper, such as blotting paper. Actually, when you prepare the with wooden boxes, Apart from the wooden boxes, newspapers also added like a sandwich. But one used the paper and replaced by another frequently. Prior to pressing it may help to immerse the brush specimen with alcoholic kills fungi. So clean with alcohol. First, when you collected the specimen from outfield, natural places, first wash it with alcohol, then only fresh it. Flesh fruit and are separated from the herbarium specimen. Place them labeled paper bag, envelope, and leave the next plant press. Fruits do not press. The keep in the readjustable pouching bags and stick it on as it is. Check and damage the paper bag frequently. Preserving aquatic plant. Aquatic plant needed immersed water, allow full expansions of branches and show habit of the plant. And preserving bryophytes and mosses. Generally, mosses, liverworts, sandworts, and lichens do not need to be pressed because already they are minute and very simple. Simply remove as much soil as possible and place each specimen in paper bag. Simple. Paper envelope and folded piece of newspaper. Split collections. Mounting. After collection, pressing and preserving, mounting. Mount the specimen that means stick on the sheet of herbarium card. The well-mounted specimen should be display both artistic and botanical qualities. It should be arranged on the card in the balanced, aesthetically pleasing way. When you apply it balancing, it is looks beautiful. Paying attention, orientation type of mounting card, arranging and attaching the specimen, position of label and accompanying slips, keeping loose parts in fragment bag. Mounting card. The herbarium specimens are mounted 29 into 43 centimeter actual quality, acid free, 29 to 43. White mounting card 2050 GSM. The direction of the card is portrait, not landscape, it should be in portrait. Arranging the specimen. If pressed correctly, the specimen should hit perfectly on the mounting card. When arranging the specimen, the leaves 1 centimeter border around the edge. Single specimen should be centered. Small plant with multiple specimens should be arranged in evenly spaced rows. Orientation of the plants in their habit. Arrange the specimens so that greatest number of morphological feature visible. Flower, one specimen, fruit, both side leaves, the specimen. Attaching the specimen. The specimen can be attached to mounting card, glying, swaying lab, although glying the stand. At the University Melbourne Herbarium Archival, white gummed tape is used for mounting. White gummed tape. White gummed tape. This is purchased in large rolls from which strips are cut at the desired length and width. Uh, Envelope, attachable label. Place it lower right corner. The attached labels place it lower right here. Scott, it's lower right. Place it 1 centimeter from one bottom right hand edges of the card. Use archival clear gum to glue it down, then to paste it. Keep the label free from mounting tape and specimen bay. Nomenclature. So anyway, after giving labeling, after collecting, preserving, storing, next mounting, label it. After mounting, label it. You should label family and scientific name, plant, including authority, collector name, date of collection, locality where the plant was collected. If also possible, also include collector specimen record number also. If it is present, record number also should write. Name of person who determined the identification, if any new. latitude and longitude, slope topography. Habits such as plant height, width, and life form. Habitat, a type of plant community including associated species. After that, namely nomenclature, author. After nomenclature, the author name of the nomenclator, collector name here. For example, the collector is the person who picked the specimen. Your entry for collector should include the first and last name and middle initial. Example, Umfari B. Beer. All answers for all collectors. Collector number. So book collector collection number also need. Collection date, locality, latitude, longitude, habitat and habit. So these are all you should include. Right. So this is herbarium preparation techniques. Just 1 minute. Right. Now biodiversity. First of all, definition. Herbarium important and also biodiversity. This is very general topic and important. First, what is biodiversity and what is conservation? This is not only important for exam point of view, and also it is useful for in our daily life, and it is your duty, fundamental duty to protect biodiversity, to conserve it. First of all, what is biodiversity? Diversity means different forms. Bio means all living organism, such as plants and animals, all together called as biodiversity. This diversity means variation in different type of plants and variation in different type of animals, and not only individuals, and also different biodiversity variation within the species also. So let's discuss one by one. Before that, let me give the brief introduction about the biodiversity. The word first given by Walter Rosen, 1985. He given the definition, the richness in variety, variable species and all living organisms in given region or habitat. So the richness variety is called as biodiversity. The other word biodiversity occurs different type of ecosystem, different type of species, organisms with the whole range their variant genes adapted different climate, environment, and of course in biodiversity indirectly influenced by the non-living things also, such as temperature, water, environment issues, all. So later we will discuss significance of biodiversity. Before that, types of biodiversity we should discuss. Uh, Types, I think, right, given. Species diversity, genetic diversity, ecosystem diversity. First of all, genetic diversity, name itself given gene, species, ecosystem, community. Right. First of all, genetic diversity. In genetic, let me take pen. Genetic diversity describe variation in number, genetic diversity, the variation in number, variation, genes, genetic, present in different species. So take different species, in the different species, variation in genes, genetic diversity. The genetic variation arises by genes and chromosome mutations, individuals and sexual reproducing organisms. That is one type of diversity. Next, species diversity. It describes variety of number. Here also same variety, number, same, and richness of the species. The species richness may be defined the number of species per unit area. Richness of species tells about the extent biodiversity site provide means comparing different sites. So this is the species level, different type of species, number richness. For example, this is area. For example, tigers, lions, or monkey. So these species are richness. So among these species, richness highest number present. So this is called richness diversity. Only three. But you take another number, another locality. If you take another locality, it has only one. Only tigers are recorded. Only tigers. So less diversity here. Here high diversity. So this is based on the richness of number. Ecosystem diversity, individual, different type of species. Describe assemblage. It referred as landscape diversity because includes size variation of ecosystem. Now we are taken not only species level, not only genus level, gene level, but here one entire ecosystem. For example, grassland ecosystem, desert ecosystem, mountain ecosystem. So you can take different type. According to Whittaker, the community diversity are three types. They are alpha, beta, gamma. The alpha, let's take pen. The alpha, this refers to number of species. Number of species in a single community. Beta describe the range of communities due to replacement of species which arise due to presence of different microhabitat, niches, different microhabitats and niches and environment condition. Range of community. It is range of community due to replacement of species. Gamma, this refers to the overall diversity and applied for the large area, both alpha. If you take, for example, in this field, alpha, only single type of species are there, single species. The beta, single species also, here also same species, but community different, land, the area different. For example, uh, this is polar region, polar region tigers are there. Here, tropical temperature tigers are there. So both the same species, but different region. That is beta. If you take the entire biosphere means the pole region, a tropical region, a desert region, tropical forest region, all together called as gamma. All together called as gamma. So this is about types of diversity. Biodiversity in India. Before that, actually in our book given uh, value of biodiversity. Value means how it is importance. What they given or not. Right. The living organism on Earth are great diversity, living diverse habitat, and processing diverse qualities. Some of the importance or values. First of all, already you know the biodiversity concept. Next, the productive value, consumptive value, social value, aesthetic value, legal value, ethical value, ecological value, economic value. These are the values or importance. In examination point of view, if they ask value of biodiversity, same question, important of biodiversity, same question. Let's say productive value. Productive, what you produce. The product, the more in biodiversity, different type of plants produces by harvesting, they can produce the commercial market, indirectly provide economical benefit to people, which include the water quality, soil protection, equalization, climate, environment, monitoring, scientific research, etc. The productive value, productive, uh, mainly not given here. For example, the food produce, timber produce commercial, food for heating, for our daily uses, the timber furniture required. So this is all productive value. Next, consumptive value. The consumptive value can be assigned by goods such as fuel, woods, leaves, forest, products, etc. And social value. The loss of biodiversity directly influence the social life of the country. Plausible through influencing ecosystem function, energy flow, biochemical cycle, This easily understood observing the determinant and effect global warming and rain, acid rain, unfavorable alteration in local process. Why it is social value means actually if there is no biodiversity, there is no recycling, there is no rain, there is no water. Simple, there is a link. That is our social responsibility. And it gives food and medicine. Already discussed that in productive. And from most of the, as biodiversity, it consists of richness of plant. Maybe these plants beneficial for food, beneficial for commercial value, beneficial for medicine, whatever it may be. And aesthetic value. Aesthetic value means the many people react positively to plants. Natural lovers, example, natural lover that are visualize appealing through the ages, poet and writers and artists from many cultures have testified to the effect of plants, beauty and human mind and emotions. Although many plants that deserve protection are unlikely to have aesthetic appeal, there are a number of frog species which can be used as symbol of conservation. This include large banyan tree, variety of carnivorous plants, giant redwood trees, animal like tiger and panda, right? And legal value. The since the earth homeland, all living organisms all have equal right to coexist. Not only human, this is this land for not only for human beings, not only for animals, not only plants, all together. Unless some legal value it is attached to biodiversity, it will not be possible to protect the rapid extinction of species. If you're not given the legal value, the proper value, there is a chance to extinct biodiversity, loss of biodiversity. Already totally biodiversity connection to one to another. For example, in biodiversity, if solar energy present, it gives the plant prepare their own food material. On the plants, they're dependent some of the herbivores. So if any disturbance occur in plant, there is no plants, there is no vegetation. Without vegetation, how can survive the vegetarians, herbivores? So when the plants down, automatically the herbivores also could be down. What happens next? When herbivores down, automatically this impact falls on the next carnivores. So like a linking chain. So ethical value. Biodiversity must be seen in light to holding the ethical value. Since man is the most intelligent amongst the living organism, it should be prime responsibility and moral obligation of man to preserve and conserve their organism, which will directly or indirectly favor the existence of the man. Just now we discussed what happen if we not focus on diversity, ecological value and economic value. And apart from that, another important value that is religious, spiritual and cultural value. From the uh, years, the many plants and animals have been spiritual significance. For example, Tulasi plant, Tulasi, we daily worship the plant Tulasi, banyan tree, they're all. The each temple consists the banyan tree and also holy plant, Osimum, Tulsi plant. For example, here in book, even Dattara flowers offered to God Shiva in northwest, but not in Telangana, in our south. Kejri plant. Kejri plant means Prosopis cinerea, used to sacrificial price. Some animal species are termed vehicle of deities, and hence including bull for Shiva, rat for Ganesh. The local communities in around forest area considered entire ecosystem. The belief, the system, many cultural places, great moral value, and the preservation of the biodiversity. It is immortal to take life and exterminate species. This is the value of judgment. This is general religious belief. Uh, the one plant I don't one region Kinjerli, right, in Maharashtra, Kinjerli. There actually the people of that area, they do not cut the plants. They never hunt the animal. Only the utilized dried plant. After plant, when the plant dried, automatically the dried sticks utilized for their daily uses. The mostly they are depend on vegetarians. Dependent, they can cultivate the crops and collected the vegetable from them only, and they can they can develop own poultry also, but they never disturb natural system. But unfortunately, in that area, the Kinjarli area, the hunted by the great hero, the famous hero Shahrukh Khan, that means Salman Khan. Till now, they believe only the Salman Khan, the poaching and hunted that area, they get jail only for that purpose, because the entire area, the holy area, the Kinjarli entire area holy. That's why in that area, each and every organism, every plant, every animal, they have been worshipping. But knowingly, unknowingly, I don't know. But he entered that area and poaching and hunting there. That's why is now taking punishment. And uses of biodiversity not given here. Yeah, biodiversity global national levels. No need. Religious scientific value. Okay. Fiber food trends. What's experts? Plant biodiversity India. No need. Some biogeographical areas in India already given here. These are biogeographical regions. Trans Himalaya, Gangatic Desert, Semi-arid zone, Western Ghats, Deccan Peninsula, Northeastern zone, coastal lands, Himalaya highlands. These are all biogeographical regions of the biodiversity hotspot. India is one of the 12 mega diversity nation of the world due to following reasons. It has 7.3% of the global fauna and 10.88% global flora as per the data collected by Ministry of Environment Forest. It has 350 different mammals, 1200 species birds, 550 different reptiles, 182 amphibians, and 4,500 plant species. It has 5,000 known species of insect, which include 1,300 butterflies, sorry, 13,000 butterflies and moths. It has 10 different biogeographical regions and 25 biotic provinces, 10 year, 10 different, these are in 10 different biogeographic region, 25 biotic provisions having variety of land of species. In addition, geographical distribution, geographical events, land mass provide high level of biogeographical diversity. Several crops arose in the country and spread throughout the world. There is wide variety of domestic animals like cows, buffalo, goats, sheep, and pigs, horses, etc. The marine biota, including seaweeds, fishes, not only seaweeds, all algal member, fishes, crustaceans, mollusks, corals, reptiles, etc. There are a number of hotspots named Eastern Ghats hotspot, Western Ghats hotspot, Northern also Himalayas, Northern Eastern Himalayas. Threats of biodiversity. After value and threats. From here given some of the areas, species also, but you can read in our book. No need to explain because Western Ghats, what type of richness plants are there, and Himalaya richness plants. So that is no need to explain. Threats of biodiversity. Another important threats value and threats most important. Value important for essay, threats also important essay. And also this is also biogeographical areas. 10 areas also important. Of course, in 10 areas you write 10 different areas, but you elaborately explain only the Himalaya region, Western Ghat, Eastern Ghats. That's it. Three spots because these are the mostly hotspot regions. Uh, Let me explain. Okay, the only three. Let me complete these three. Right. For example, this is India. These are the Western Ghats. Western Ghats. Trans-Himalaya regions. These are Trans-Himalaya, Northeast, Northern Ghats. These are. First, let's see Trans-Himalayas, Himalaya region. The Indian Himalayas are classified into Western and Eastern Central Himalaya, which is located in Nepal. In these, they are all in the Nepal. There are different type of Sikkim, Darjeeling, Arunachal Pradesh, Himachal Pradesh. They are all. Mainly, the Western Himalayas are seen with the tropical, subtropical, temperate forest. Pinus, mainly it has the Pinus species. Cedrus, Pinus and Cedrus, Abies. Most of the gymnosperms found at that area. Eastern Himalaya cover tropical evergreen. Eastern Himalaya, tropical evergreen, tropical evergreen, tropical evergreen, tropical grassland, subtropical temperate, subalpine forest. Here the mainly Dipterocarpus, Shoria robusta, some ferns and lichens are found in that area. Desert, semi-desert, Western Ghats. Another important hotspot is Western Ghat here. The extent 15,000 kilometer here from till year to year. From Tapati to Kanyakumari. Here from Tapati, Kanyakumari. Tapati to Kanyakumari. The altitude range from 200 to 2,700. Rainfall up to 700 centimeter here. Here in that area mainly sholas. The remarkable feature of Western Ghat is the presence of sholas, shola trees, which present remnant patches evergreen vegetation along the folds rolling down of the hill Nilagiri. The famous Silent Valley. These mainly consist of medicinal plants. The Agasthyamala are located in this zone. Some important species, Beauty monosperma. dry deciduous forest, all dry deciduous forest, semi-evergreen forest, they are all present in that area. Northeast zone, Brahmaputra Valley, Northeast Hills, it covers the steppes Northeast India except Arunachal Pradesh. It is one of the richest biodiversity hotspot in India, 8,000 species. Northeast zone, richest biodiversity, remember it. So these are the different type of winds. Shola, that means evergreen plants, sholas. Right. Threats biodiversity is considered as a reservoir resources to be used manufacture food, medicine, industrial already. Value means importance. How it is value? We give value, it gives the food and medicine and it gives the aesthetic value, commercial value. That's why we should protect. But with an increased demand of rapid population growth, biodiversity is gradually depleting, gradually decreasing. Number of plants and number of animals extincted by many endangered. Let's see the what are the factors for extinction. They are the considered threats. First of all, habitat destruction. This is the main. The primary cause, loss of biodiversity, the destruction, which is resulted due to the large industrial and commercial activities isolated with agriculture, irrigation, construction, dams, mining, fishing. So one of the best example, the habitat construction recently, Papikondalu, which is located Andhra Bhadrachalam region here. So the government decided to construct a project there so that they had to remove some region, Papikondalu. But due to the construction of the new dams, due to new check dams like that, the Papikondalu region, already it has the rich biodiversity. When you remove this area, almost the loss of the biodiversity. And another one is admit fragmentation. Fragmentation means there is no completely separate. In habitat destruction in that particular area, for example, if you take for example, this is Papikondalu area. This is all the high richness. As the government decided to plan here the construction a dam in this region, so that they can remove this area. Already it has the different type of variety of biodiversity there. Now this entire location destroyed. So the area organisms are destroyed and it replaced by dams. Fragmentation. With increased population, habitat are fragmented into pieces by road. Already here the one forest area it is. So now the government want to planning to road map. When it is interrupted, this region break into pieces. That mean, for example, here the mountain like a separated like this. And here species are there and here species, but here the road. In that situation, the automatically when the road fields, canals, not only roads, fields, canal, power lines, town also. The isolated fragment of habitat restricted to the potential species for dispersal and colonization. In addition, the habitat fragmentation also bring about microclimate changes in light, temperature, wind, etc. That means why it is not good for the biodiversity because already here the species separated by the canal. So any interrupted region, but here some animals are there, here some animal there. So it is dispersal of the seeds, and also not only seeds and physical contact of the animals also not possible. The lack of mating, the production gradually decreasing. This is likewise the habitat loss. So this is second one. Third, pollution. The most dreaded factor inducing loss of biodiversity environment pollution, which include pollution, water pollution, maybe industrial pollution, chemical pesticides, radioactive material. Over exploitation. The natural resources are over exploited to meet growing rural poverty, intensive technology growth, globalization of economy. All these factors together, many responsible for the extinction number of species. So, uh, and exploitation, another important, here given or not, I think factors. Let's category tundra, flood created flora, fauna. Right. So over exploitation means for most of the people for fun, enjoyment, they poaching the plants. So poaching the animal, hunting the animal for their fun and enjoyment. Due to the fun and enjoyment, they are exploited. And for the growing people need commercial benefits. For their benefits also, they depend on plants. While growing people depend on plants, automatically the plants or whatever may be, the plant or animal, while as they depend on that, the gradually decreasing number. Actually, the diversity, whether plant or animal, have been not growing as grow as animals, as grow as human beings. So that's why it is when they depend on this diversity organisms, they gradually decrease. And another decrease of the biodiversity, exotic species. The introduction of exotic species are due to the horticulture, agriculture, European colonization, accidental transport. One of the best example, Sarkaru plant, actually not found before when they are introduced from the Australia country. Now when they are very rapidly, rapidly distributed. And when they are rapidly distributed, automatically this effect on other plant and also weed plant also. When you planting a crop in that crop area, instead of crop plants, the weed plants will grow more. And diseases due to vulnerable infection, some anthropologic activities may increase incidence disease. So due to the disease also, the existing disease. So for example, the influenza virus, which is found in birds, automatically the birds genera completely decrease when it is infected. Due to the flu, especially bird flus, due to flu, the population completely decreasing. And shifting to Jammu cultivation, poaching of wildlife already completed. Endangered and endemic species India. Given higher plant is to enlisted 15,000 mammals, reptiles, birds, fishes. Now, what are the strategy for conservation? Okay, these are the threats and values, right? How they conserved? You're given plants factors of threats following the key factor: harvesting timber as logs, poles, charcoal, expanding farms, exploitation, uncontrolled grazing, livestock, tourism and other visitor pressure, mineral oil extraction, developmental program like construction, dams, already told you, habitat destruction. forest, environment, pollution. So they are all completed. No new one. Uh, Threats categorized in threatened flora. Before that, that mean extinct, endangered, vulnerable. Threat category: extinct, already not present, endangered, near future, vulnerable. It takes some more time. Rare species. So that is the based on the this presence of the plant species. Now conservation of biodiversity, in situ conservation and ex situ conservation. Name itself given, in situ conservation of biodiversity is the conservation species within the natural area. That is important. Within the natural area. Within their natural area, if they protected, that is called in situ. In this method, natural ecosystem maintained and protected. The in situ conservation has several advantages. Following the important advantage: cost effective and convenient method converging biodiversity. That means within the area you protected. As a natural place, it includes a number of organisms. You can conserve much more number of organisms. Since the organisms are natural ecosystem, they can evolve better and can easily adjust different environment condition. Certain project area in situ conservation takes place include national parks, wildlife sanctuaries, and biosphere reserves. These are all one type of in situ. First of all, let's take national park. These are a small reserve maintained by the government. Small reserves. National parks, they are the small reserves maintained by the government. These boundaries are well demarcated and human activities such as grazing, forestry, habitat cultivation prohibited, strictly prohibited in the national park. Example, Kanha National Park, Bandipur National Park. Next, wildlife sanctuaries. These are the region where only wild animals are found. Boundaries are demarcated. Sorry. Wildlife sanctuaries. These are the region where only wildlife already existed. A wildlife protected. Human activities such as timber harvesting, cultivation, collection of food, and other forest products are allowed. So in wildlife sanctuaries, in first in national parks not allowed, but in wildlife sanctuary, human activities, timber harvesting, cultivation, collection of wood and other forest products are allowed here as long as do not interfere with conservation project. Also, tourists visit this place for recreation, wildlife sanctuaries. Biosphere reserves are multi-purpose protected areas, whereas the wildlife, traditional lifestyles, inhabitants, and domesticated plants, animals are protected. Tourist research activities permitted here. Here also same. The purely restricted national park. Wildlife permission, biosphere also permitted. Exit conservation outside. Exit conservation biodiversity involves the breeding maintenance of the endangered species in artificial ecosystem, such as zoo park, nursery, botanical garden, gene bank. There is less competition for food, water space among the organisms. The executive considers following advantage. The animals are provided with longer time and breeding activity. So that means improve population. The species breed capacity can reintroduced in the wild. Genetic technique can be used in the preservation of endangered species. First, botanical gardens. Initially, botanical gardens were created as aesthetic region where different plants were grown for pleasure. However, the period of the botanical garden have become center for scientific research for propagation of many local and exotic plants. Zoological park, zoo parks are places where the animals are kept in captivity. Many such animals are rare or endangered and many cases extinct in their natural habitat. The zoos plays an important role in the breeding of rare endangered species. Sometime after successful breeding, many species are released in their natural environment. Thus, the zoos are major contributor in checking the extinction of the endangered species. The zoos always significant contribution creating awareness and interest in livelihood space among the children. Gene bank, seed bank for future uses. We collect the seeds from the different plants. We protect and store it for future uses, future purpose. Tissue bank, same like that genes. The tissue collected from the explant, the conserved, stored in the lab. Like cryopreservation, that is cryopreservation means the newest application technology for preserving of biotic part, whether it is germplasm, seed bank, tissues also. Their part are stored liquid nitrogen minus 196 degree centigrade. The metabolic activities of the organism are suspended under low temperature, which are later used for research purpose. So this is biodiversity, in situ conservation, botanical garden, zoological juice bar, national international conservation strategy. Actually not given. National international strategy. There are many organizations in the world who conserve the biodiversity conservation. The intergovernment organization like United, where it is right. Here are the conservation strategies. Conservation strategies. So there are many organizations in the strategy that is first of all UNEP. United National Environment Program. It is a governmental organization like Ministry of Environment Forest. Another one is Ministry of Government. WWF, World Wildlife Fund, and UNCED, UNCED. The most important contribution from international framework in conserving biodiversity is the United Nations Conference, United Nations Conference Environment and Development, famously known as Earth Summit. It is famous known Earth Summit. Held in 1992 June, Rio de Janeiro, Brazil, UNCED, famously known as Earth Summit. That is what is UNCED, United Nations Conference on this is full form important. United uh, United Nations Conference, United Nations Conference on Environment and Development. Environment and Development. Other important international conference biodiversity are CITES, TES, TR, TRA, FFIC, CITES, Convention on International Trade in Endangered Species, Convention on International Trade in uh, Convention on International Trade on Endangered Species, site. Traffic, another one is traffic, trade, record, analysis of flora and fauna. trade record analysis of flora and fauna. Of, of flora and fauna. So these are the national international conservation strategies. And now so far, anatomy. The online classes start with anatomy part. The anatomy completed, entire anatomy completed, and next started the taxonomy part. And taxonomy portion also completed. And in this book, Gymnosperms, taxonomy of Angiosperms and anatomy. Gymnosperms part already completed offline mode. The offline mode, the entire Gymnosperms completed. So in online mode, taxonomy of angiosperm completed, anatomy of completed. So the complete C3 books completed. And now gymnosperms. In gymnosperms, important topics, character, general character of gymnosperms, economic importance of gymnosperms. If you learn general character, the same concept you can apply for the type studies. The or type studies, what they given orders, I think, of coniferales, needles. So like the if the coniferales are in coniferales, uh, pinus, cedrus. So different type studies there. So if you take any one, the almost similar character, all follow the similar character. There, if you when you compare with cedrus or pinus, just about 10 to 20% difference. The rest of the characters are same. That's why what I'm telling, the just focus on general character of gymnosperms. If you learn general character in examination point of view, what they given, you should write. You can write any type study. For example, you know gymnosperm, we don't know about pinus, but examination point of view, if they ask pinus, you just write a general character. That means generalize the type study. But if you write like that, instead of, for example, 10 marks, instead of 10 marks, you can get 5 marks. So that is the easy strategy to get the more marks. What you don't, even you don't know the question, you can write the answer based on the general character. That is the only one concept to get marks from the gymnosperms area. And anatomy area, the anatomy area also, the only one key factor is the all internal structures, epidermis, cortex, vascular bundle. If you take stem, if you take root, same internal organ, same organ, but somewhere variation in vascular bundle only. Only vascular bundle changes. That one here also same in anatomy. If you learn some one stem or root, whatever it may be, if you get one, if you learn one, you get more. That is from anatomy side. But it's very difficult in taxonomy. Very difficult in taxonomy. Because in taxonomy, you should read ICBN. Now recently called ICN, the entire strategies, rules and regulation of ICN. Later, Bentham and Hooker classification important. Engler and Planter classification important. Hutchinson classification important. And recent trends in taxonomy, chemotaxonomy, numerical taxonomy, paleotaxonomy, embryo taxonomy. So the like that all different taxonomies are important. There is no relations to one question to another question. But in gymnosperms, one topic relate to other topic. They are joint. In anatomy also same. But in whereas in gymnosperms, no relations ICN, rules and regulation different. Hutchinson classification different. Bentham and Hooker classification, Engler and Plantel classification. But you can compare. You can compare, you can differentiate with that. And in important thing is in taxonomy, another one is taxonomic order. Already, when I explained it to taxonomic order, poales or poales, Amentiferae, different type of orders, and tubiflorae. Just remember the families. What are the families in that particular series, in particular order? That families, for example, if you take tubiflorae, more than 10 families are there. 10 families, character similar. Here only when enter the taxonomic study, taxonomy, in order level, family level, when enter that, there the similarities occurs. For example, gamma petalic, all gamma petals similar character. Petals fused, epipetalous condition. Already clearly explained with differences, comparison, with comparison. When I explained the previous classes, once again go back and watch it. I clearly explained it with differences, one family to another family within the order. So that is the important. That is the way of studying to get marks. And this is not only for theory and also for CSR, NET and AP set preparation also. This is all important. Okay, all the best one and all. Thank you.